All right, welcome everyone to the April edition of the IPM Hour. Today our speaker is Dr. Cynthia Ocamb from Oregon State University, where she's an associate professor in the Department of Botany and Plant Pathology. She did her undergraduate work and master's work both at North Carolina State University in horticultural sciences and plant pathology. Cynthia, you must have been happy about March Madness this year, or relatively happy, I guess, right? Um, and a PhD at University of Minnesota in plant pathology as well. She worked in the Forest Service in Minnesota for about 6 years before joining OSU. Her program focuses on the diagnostics and diagnosis and management of diseases in field and vegetable crops. And this includes training of county-based faculty, crop consultants, and producers on disease symptoms. And this was really impressive to me, and I don't know if this is all still right, but she is currently the section editor for Plant Disease Management Reports, senior editor for the journal Plants, and co-editor of the Pacific Northwest Disease Management Handbook and website. So it's like, holy moly, that's a lot of work. And today, Dr. Ocamb is going to talk to us about powdery mildew in hemp.
Thank you, Matt. I must say my information on the website must be a little bit out of date. I did drop the editorship of Plants because it was all becoming too much.
Yeah, I'll bet.
Yes. So I'm going to go ahead and start sharing my screen. And hopefully everybody can see that just fine. And thank you for everybody of your attention today for this presentation. So this is a large team project. It's not just myself. My co-PI David Gent and Hannah Riverdale have both played major roles in this work that I'm going to talk about today, as well as Andrea Garfinkel from Oregon CBD. And then we have junior scientists Brianna Clausen, Michelle Weissman, Steve Massey from Dave Gent's project, and then Bill Thomas from mine, who are also made major contributions to the information today. So just to give you some background on generalities of hemp and hop production. So hemp and hop are the two economically important crops in the family Cannabaceae, and hemp is an emerging crop across the US and including in the Pacific Northwest. And so because it's such a young crop in the relative scheme of things, there's not a lot of information that is known about the disease aspects, especially in the Pacific Northwest. And then the hop production has a very strong focus in the Pacific Northwest. So Washington State leads with the acreage, followed by Idaho and then Oregon. And between the three states, about 98% of the hop acreage is based in the Pacific Northwest. And powdery mildew has been a long-term problem on hop in the Northwest after it showed up in 1997, first in Washington State, and then the following year in Oregon and Idaho. So Podosphaera macularis has been long studied. I worked on it some, and then when Dave Gent joined the USDA, he took over the powdery mildew program. And so there's quite a bit of information that's been developed on the powdery mildew on hop. And we know that the powdery mildew overwinters on infected crown buds. And here in the photograph, this is what we term a flag shoot, where the Podosphaera had infected a bud the previous season. And then in the springtime, when the bud started to grow, if the mycelium survived the winter, then it would be able to continue to grow and then start to reproduce on the surface of these tiny shoots. So this flag shoot is typically the way that the powdery mildew Podosphaera overwinters in the Pacific Northwest, because we only have one mating type at this time. You need two mating types to complement each other in order to get the sexual reproduction and produce the ascospores in the springtime. But right now, everything is a conidial-based system in the hop powdery mildew. And so from these infected flag shoots, you'll get secondary spread onto leaves of the plants. They're typically growing on a tall trellis, and you'll have quite a bit of leaf infection that can occur, especially on certain varieties. And this is a Zeus plant in the greenhouse, so you can see how much disease can develop on the leaves. And then the economic impact really comes from infection of hop cones by Podosphaera. So infections of the cone can occur throughout the cone life, it's believed. And when the cones are infected young, you'll get a distortion of the cone shape, such as you can see right here and here. And the cones can also take on a brown coloration for certain varieties. So Willamette had issues with powdery mildew infections being associated with cone browning, which resulted in tremendous losses when brewers would reject certain harvest lots of the Willamette cones. So that's just sort of a general background on the powdery mildew, Podosphaera, and hop. And then Golovinomyces ambrosiae has been reported on powdery mildew, on hemp in the Pacific Northwest. And Michelle Weissman took the lead on reporting that. She's with Dave Gent's lab. And these are photographs of infected hop leaves that I took last summer. So here's a very small infection started here on this leaflet of a hemp leaf. And these are very difficult to see in the field at the early stage if you don't have practiced powdery mildew eyes. And then this is a leaflet set where you can see the powdery mildew has expanded a little bit further, so the Golovinomyces is pretty apparent at this point. And then it can spread quite considerably depending on the variety, perhaps, and the environmental conditions. And so this is a plant that I saw in one field site last summer, and you can see that there's extensive colonization of the hemp surface area. So it's also reported to be able to infect a number of other plants, the asters, the ragweeds, dahlia, zinnias. Now, there was a paper that came out in 2020 that I looked at recently that talked about Golovinomyces, including the ambrosiae, and they looked at the genetics and the relatedness of several species within the Golovinomyces and that were obtained from different hosts. But unfortunately, that paper didn't include hemp. So I have some questions in my head about the isolates that grow on hemp. Will they infect these hosts? And that's something that I would like to look at down the road. But right now, my assumption is that this powdery mildew species can have other hosts beyond hemp. And that's partly due to the fact that we haven't seen Chasmothecia production on hemp has not been reported yet in the US. So it may be due to that there's just one mating type present. We don't quite understand yet. And in 2020, I had a project underway on the botany farm with a former graduate student, Taylor Ashton Bates. And we were looking at botrytis control on hemp in the field just for one of those spray trials, the simple spray trial. But the hemp field came down with powdery mildew. We saw that in, I think it was September, that it started to become obvious that I noticed it out there. And so this is a photograph of one of the branches that has buds on it of one of our hemp plants. And you can see the white colonies that have formed on the bud material, and they also were on leaf material. And it started towards the center of the plant, and it was confirmed to be Podosphaera macularis by Dave Gent's group. So this led Dave and I to put together a grant proposal for the Western Region IPM initiation project program, looking at the powdery mildew risk with hemp production in the Pacific Northwest. So it had 4 objectives, and the first one is mainly what I'm going to talk about today, was just looking at the extent of powdery mildew occurrence on hemp. And then Dave's group was characterizing the fungi that we recovered. They were categorizing by species, mating type, and then some virulence aspects for the Podosphaera type. And then we also have work underway in the greenhouse that's been started about a month ago, trying to screen hemp lines for susceptibility to powdery mildew. And then, of course, communication of results to the stakeholders. So if we look at the survey with the hemp fields for the powdery mildew, we examined in 2021 32 fields. 21 of those were in Oregon. So this is a Google Earth map showing with the yellow pins or the golden pins of the different locations where we had hemp fields. I'm going to confuse hemp and hop. I'll be very careful with that though. So the hemp fields were focused in the Willamette Valley, and that was intentional because we were interested in having fields that would be in the vicinity of hop fields potentially, so that we could look at this interface of where long established hop production also had new hemp production. But we did have some fields located towards the southern part of the state. So we had a couple around Roseburg, and then we had one around Medford. And then we had these couple up here that are really outside of the Willamette Valley, up in Troutdale along when you're getting close to 205. And then for Washington, we had 11 fields that were located in the Yakima Valley. And so Yakima Valley in central Washington is the major focus of hop production in Washington state. So they ranged across the valley, but we feel like we have a good representation of the interface of hop and hemp production in the survey. So the methods were that we had 3 time points that we went out and examined plants: early, shortly after transplanting, mid-season, and late season. So Andrea Garfinkel here in this top photograph, you can see how tall the hemp plants were at late season. So this is Hannah and myself in the lower photograph, and we were also struggling to do this between raindrops because it's difficult to evaluate powdery mildew on wet leaf material. So it was a challenge, the late season work. I greatly preferred the early season when the plants were about 1 foot tall. It was much easier to look at these. But in the survey basically involved looking at 2 transects of 100 plants. If the field was small, 20 or less rows, then we looked at 1 transect. And then on each plant, we evaluated 10 leaves for the presence of powdery mildew. So what we would do is we would put a person on either side of the row and then go down, 100 plants, and then look at stratified areas, randomly blindly choosing a leaf to evaluate for powdery mildew. And one of the things I learned too is when you're leaning into hemp plants that have a lot of sticky buds, you can actually etch your plastic of your sunglasses. So one of the things I learned. And if we detected powdery mildew in that hemp field, then we collected up to 9 leaf samples to bring back to the laboratory. And then the powdery mildew mycelium and spores were lifted off with a sticker tape system where you take a little sticker, a little piece of cellophane tape, and you dab it on the infected colony area or the colony area, put it in a microfuge tube, then we stored it in the freezer, and then those went over to Gent's lab for characterizing. And his laboratory work focused on the characterization of the fungi. So they did that with the 28S to determine the fungal species. And then they also, for Podosphaera, looked at the mating type and some of the potential virulence genes that could be in place, just to get an idea of the genetic construct of the Podosphaera. So for the survey results, this is the map of both areas, the Oregon fields and the Washington fields. And the purple pins signify where Golovinomyces was detected. So we have 12 fields in Oregon. One of them is hidden down here behind the gold pin around Roseburg. And then one of the other pins up here represents 2 fields that were side by side. And then there was 1 field in Washington in the Yakima Valley where the Golovinomyces was detected. That sample was actually came in after the last survey point, but the producer noticed it, and so they sent in a citizen science sample to Gent's lab. So we did get a powdery mildew up there from Washington that way. And we also though found that Podosphaera macularis co-occurred at several of these fields. So these are signified with the red pins. The purple pins are the sites where only Golovinomyces was detected, and the red pins is where both of the fungi both genera were detected. So we can see that one of the Troutdale sites had Podosphaera as well as Golovinomyces. And then there was one here in the heart of the hop production in the Willamette Valley that had both. Another one down here, starting to get to the south edge, but it's still part of the Willamette Valley. And then one of the fields down there by Roseburg had both of the fungi. And the field up in Washington had Podosphaera as well as the Golovinomyces. So just to look at the incidence of the powdery mildew, and these are numbers that are based on generally 2 transects of 100 plants, 10 leaves. So this would represent the percentage of 2,000 leaves, unless it has an asterisk over here, and that indicates that the field only had 1 transect that was surveyed. So we can see that there was quite a range in terms of the incidence of leaves with powdery mildew in the survey. This is the last survey date that I'm showing here, because this is when we basically were finding it. So it ranged from close to 70% incidence to down to less than 1%. And it didn't seem to matter in terms of the field size, because I was expecting that the smaller fields would maybe have, less likelihood of powdery mildew, but that didn't seem to be the case. And then we did find 2 of these fields in Oregon had powdery mildew by the mid-season stage, so that we first picked it up. And both of them were ones that had higher levels by the late season evaluation, which makes sense. So here you can just see the pluses for where they were all Golovinomyces positive, and then the ones that had the Podosphaera detection also. And then in Washington, they're all zeros. So I left the zeros in here because it doesn't confuse the eye. And again, this one detection was picked up after the 3rd survey date, but before harvest. So we were able to get some samples sent down to Dave's lab where they could detect that both fungi were occurring. So for a summary, it seems that in 2021, powdery mildew showed up late in the season in our survey, but also the powdery mildew on hop was very low in 2021. So it's expected that the high temperatures that occurred early in the summer of last year in June, we had like 115, 117 degrees. We had a few other heat snaps through the summertime, and it just generally seemed that powdery mildew levels were lower overall. So I'm not positive that we're going to redo this survey this year, because we have additional funding to do a 2nd year. And I'm not positive that it's going to always be showing up late in the season. So we'll find out hopefully this year. And both Golovinomyces and Podosphaera can infect hemp fields naturally in the Pacific Northwest. So this will be something that the hemp growers may have some concern about for their production, but also may have a larger impact on the hop growers. We don't know at this point. This is a first report for the Podosphaera macularis infecting hemp in Washington. So that's significant. And then also in Oregon, the Podosphaera was detected on hemp outside of the Willamette Valley, what I consider generally the big hop production region. But I did look in the look around to see if I could find any information this week on hop production in the southern part of the state, because it used to be there. It used to have hop production down into California, I believe, a number of decades ago. And there is one hop farm that I did find registered that is located just north of where that infection was in the southern part of the state. So Roseburg, just north of Roseburg, there is one hop farm. So it could be that the Podosphaera is going to be associated with hemp when the hemp is grown in the vicinity where there is also hop production. But I think that this offers some concern in the big picture about having hop and hemp in close vicinity. So this is a photograph of one of the fields in the Willamette Valley, where you can see here's hemp production right here. These are just rows of hemp. Over here is a hop yard. you can see the wires and you can see the trellis, but these hop plants have been harvested because this was our third evaluation date. So generally the hops are harvested before the hemp is harvested, but they can be in close enough proximity that they could have an exchange, obviously, of Podosphaera inoculum between these two crops. And this could be a potentially an issue for hemp, getting an earlier infection of Podosphaera, say by mid-season or even earlier, because the hop powdery mildew will be active starting in April, May. And so by the time that these plants are transplanted in the field, there could be inoculum pressure right next door. And the other aspect is that when you have a hemp field and then you have the hop field that's been harvested, the hop field is going to start to regrow in the fall. So this fall regrowth is very normal. It's part of the hop physiology. It just wants to grow and grow and grow until the day length gets really short, and then it will quit growing. And you'll see that in the greenhouse too. It's difficult to grow in the greenhouse sometimes. But with this fall regrowth being present before the hemp is harvested, potentially there could be some movement of the Podosphaera from the hemp field back onto the hop yard. And we're not quite sure of what kind of mixing possibilities could occur here genetically. So this does, I think, raise the concern about, is there potential for development of novel genotypes by Podosphaera macularis that could increase the powdery mildew pressure on hop plants? And vice versa, will we see with these two crops in vicinity that just the mixing back and forth would just bring up new strains that could be more virulent or cause more severe disease or overcome resistance? So I want to acknowledge, first off, the hop producers who collaborated with us and allowed us to come out into their fields multiple times and had to deal with us calling, et cetera. They deserve a huge thank you. Also, support was provided by Oregon State University and the USDA ARS to allow Dave and I and others, time and space, et cetera, to do the work. And then, of course, there was funding that was provided by the Western Integrated Pest Management Center through their 2021 annual grant program. Thank you very much for that. And then we had assistance with the sampling beyond the first slide of all the people's names. We had assistance provided by other members. So some of the undergraduates from my lab, Darby, Ashley, and Anna, were extremely valuable with helping out with the survey, as well as Brooke from Oregon CVD. So with that, this is sort of a short talk, but if there's any questions, I'd be happy to try to answer any questions that you may have or points you want to discuss.
Well, thanks, Cynthia. So for everyone, you can since this is not like I think it is a webinar, but the format of it does allow people to take their mics off of mute. So if you would like to do that, you're welcome to do that. Or if you like, you can also type your question in the chat box. So we're monitoring the chat box. I'm not seeing, or you can raise your hand too, I guess, if you decide you want to do that. I'm not seeing any questions yet. Actually, I have a question. The uspest.org website is produced by Oregon State University through the IPM program, and it models a whole variety of different crop types and pest species. And there is a model for hop powdery mildew in that suite of models. And I was kind of curious, considering that the Podosphaera as well as this other genus both occur, well, at least the Podosphaera occurs in both, right? I'm guessing that how did you pronounce that other one?
Golovinomyces.
Golovinomyces.
Just say it fast.
Yeah, Golovinomyces. I'm assuming that one doesn't occur in hop, right?
There's been some work done with it that shows that you can start to get some initial mycelial colonization on the surface of hop, detached hop leaves, as I think how it goes, but you don't see much sporulation. And then it seems like when you inoculate an intact hop plant, that you don't see infections at that point. And Dave can correct me if I'm wrong.
So I guess my question is, could we use the models in the uspest.org to predict what's likely to happen with, I mean, would that model be useful for us to be able to predict what's likely to happen with the two powdery mildew genera in hemp?
You know, that's a good question. I would think that the Podosphaera could be modeled by the hop model, because that goes back to environmental conditions. Of course, there would need to be some double checking to make sure that the upper temperature limit, which I think is part of the model, is the same for hop as hemp, because that could maybe be slightly different, because the hemp plants may offer more shaded inside of the plant, because the plants tend to be a little bit more open than hemp plants that are trained on the strings. But I would think that ultimately it wouldn't take much to convert that model from hop to hemp to include hemp. Now the Golovinomyces, I'm not positive. I think we have to look at the environmental conditions, the constraints under which that fungus would reproduce on hemp plants to see if it, because I would say looking at it superficially, it seems like the Golovinomyces is much more able to colonize leaves that are in full light exposure right off the bat, where I have a sort of a stereotypical take on Podosphaera that it likes to infect inside the shaded area first, and then when there's a lot of sporulation abundant, then you see it on the outside of the plant. So with Golovinomyces last summer, I could see that sometimes the powdery mildew, when it was just starting, wasn't limited to inside the plant. So there could be some slight differences.
I get you. Yeah. Okay. So there's a question in the chat from Len Coop, which is, what does the Oregon State University Global Hemp Innovation Center, or what is the Global Hemp Center doing about supporting powdery mildew control and breeding for resistance?
Yeah, so Leonard, that's a good question. And what I would say is I'm working in concert with the Global Hemp Center now. And so, and also in concert with the USDA, with Hannah Riverdale, so that we're official collaborators, and we have a DACA agreement with the USDA. So I would say that in the big picture, we're getting support to understand the disease better. This survey this past summer was really our first attempt to get out there and see how much was really out in commercial production. And then I'm collaborating with the Global Hemp Innovation Center to look at powdery mildew resistance in lines, so that they're assembling a germplasm collection. And then I'm participating with screenings of that material initially for powdery mildew, and then probably other pathogens as we progress through the next few years.
Yeah. Okay, we have a second question from Steve Elliott. Are there label restrictions for fungicides for either crop, hemp or hop, that affect the use in the other? And a second part is, overuse or resistance becoming more likely?
That's a good question, Steve. So what I would say first off is, if we talk about hop, the hop industry in the Pacific Northwest generally has a really big set of fungicides that are registered for use on hop for powdery mildew management. And there's a wide ranging FRAC group representation in those labels that are registered. But yes, we do see in the hop industry that there can be a tendency. Originally, many years ago when I was first doing powdery mildew before Dave came on board, that there was a tendency to want to focus on one or two active ingredients because of the price. But I think it's been successful over the past 20 years that the growers understand the risk for developing fungicide resistance, so that they have embraced the idea of rotations of active ingredients or tank mixing active ingredients, so that basically there's a resistance management program on these farms, because they recognize the devastating impacts that powdery mildew can have. But when you look at the hemp industry, there's not nearly the fungicides that are labeled for hemp. And I noticed this a number of years ago when our governor, Kitzhaber, pronounced that hemp could be legally produced. So I was doing my job as an extension specialist in plant pathology, that when chemical companies would come to my office and visit, that I would bring up, these were potential problems if we have a hemp industry. These are some of the diseases that we could be concerned about, and including having fungicide, seed treatments, et cetera. And generally, back about 8 years ago, I could feel a lot of resistance, that was said out loud by the company reps, and they tended to be large companies. And that their attitude was, they didn't want to go forward with registrations for cannabis, because if you go for cannabis, you're not just dealing with hemp, you're dealing with marijuana, so some of it could be cultural things, but also I think the smokable product offers a lot of different issues for fungicide registrations. So there's not a ton of materials that are registered for fungicide applications to hemp. I think the potassium bicarbonates and the oils are the main ones that are registered for hemp right now. I'm not as concerned about the problem with developing resistance with those, since they're, the bicarbonates, the contact active material, et cetera. But I think as we start to get fungicides registered for hemp, for example, if powdery mildew seems to be a target that companies are interested in going after, there could be an issue if we only have 1 or 2 frat groups introduced that are more traditional chemistries. Then I think we could have problems with overuse and resistance developing. So that could be then translate into a problem for the hop growers, because there could be a population of Podosphaera resistant, fungicide resistant Podosphaera strains that develop on hemp that could affect hop.
Yeah, makes sense. Okay.
I have one more question from Leonard Coop. That are you restricted from studying powdery mildew on marijuana due to still being nationally illegal? Do you think the issues would be the same? Yes, I am restricted. I do not work on marijuana. I only work on hemp that is, certified to be hemp. I've turned down looking at germplasm that's unknown about the THC level. I'm like, I just don't want to get into that whole. It seems like just a messy thing to get involved in. So I'm not working on marijuana. If the federal government, if the Congress votes to change that whole situation, then that might change the equation for me. But I do think that the issues in terms of Podosphaera attacking marijuana type cannabis, yes, I think that could be an issue for marijuana. I don't know if I would say the issues would be the same, because I expect that there's going to be a strong influence as we go through the years and have Podosphaera have more adaptation for hemp. The genetics that are present in the marijuana are likely quite different than what are present in hemp. So I don't know about that arena. But in terms of the pathogen, potentially being able to infect and having to consider doing management strategies. If we see it a problem on hemp, I think it'll be a problem on cannabis, and then it could be even, marijuana, and it could be a bigger problem on marijuana in greenhouse production, because I think the greenhouse conditions could really foster really high levels of powdery mildew.
Yeah, makes sense. Not seeing any additional questions. Thanks again, Cynthia, for a really great talk. That was marvelous. I just want to remind everyone that our next IPM hour is coming up May 11th, and that's going to be Ashley Larson, who's going to be talking about pesticide use in organic fields. Steve, is there anything else I've forgotten?
No, I don't think so.
Okie dokie. Thanks again, everyone, for joining, and we will
Thanks for
see
your
you
time.
next month.
Bye.